Good morning, children. Yesterday we did about the Golgi complex, and in the Golgi complex, we studied that. I'll just give you a review of that. The structure it was studied by, or it was discovered by Camillo Golgi, the nerve cell of cat and barn owl. Camillo Golgi. Camillo Golgi. He discovered, he observed it in the nerve cell of cat, cats, and barn owl. Here he studied later on the ultrastructure of that, or electron microscopic structure, or ultrastructure. It was Golgi body was studied by Dalton. The Golgi, this is what I taught you. The Golgi body is known as dictyosomes in plants. Dictyosomes in plants. This was another point which I gave you. Then the next point I told you about the size. And in size, I told you that if the cells are secretory in nature, secretory cells like hepatocytes which secrete bile, number one, like hepatocytes. Hepatocytes are liver cells, and they are responsible for secreting bile juice. They secrete bile juice. Then we have another cells like the pancreatic cell. Pancreatic cells, and these cells, they what do? They secrete pancreatic juice. There, these Golgi bodies are larger in size. In these type of cells, they are larger in size. When this type of condition is seen in the secretory cells, they are larger in size. Larger in size. Whereas if the cells are non-secretory, in non-secretory cells, that means which do not secrete anything like muscle cells. There, the size is small, so smaller in size. There they are smaller in size if the cells are non-secretory in nature. Then what I told you about the number, their number, this was about the size. Now, then I told you about their number, and in their number, I told you that in if it is animal cell, in animal cells, the number are few, maybe one or two in number, and if it is plant cell, the numbers are more. That means it can be from 6 to 10. So the numbers in plants and animal cells are different. Then I told you that in animal cells, the position, and in plant cells, the position. Their position of Golgi body. All this I'm teaching you about Golgi bodies. So if it is position also different. If it is animal cell, in animal cell, the position is perinuclear. Perinuclear means near the nucleus or close to the nucleus. The position is close to the nucleus in case of animal cell. And if it is a plant cell, then the position is different. It is scattered or diffused. In case of plant cells, if it is a plant cell, the Golgi complex is scattered, or we can also say diffused. Scattered or diffused in case of plant cell. Then I told you one more point, zone of exclusion. And what is this zone of exclusion? Is the clear cytoplasm complex or Golgi and then one more point which I shape according to the function. This is what I all the characteristics, the general introduction of Golgi complex or Golgi body. Now I move on to the next point, and in which I tell you the structure of Golgi complex, which the ultrastructure or the electron microscopic structure, which was given by Dalton. This structure I'm going to. So let's come to the second point. Let's come to the second point. The ultrasound, and it was given by Dalton. According to this structure, there are three components of Golgi body. It has three components. And the three components of Golgi body are, it has three components, and the three components of Golgi body are cisternae, vesicles, and vacuoles. Number one is cisternae. Number two is vesicles. And number three is vacuoles. The three components of Golgi bodies are these. Now, if we see the ultrastructure, we find it like this. That is one cisternae, second cisternae, the third cisternae, and the fourth one or solon. Here there is no space, so I'm just making it like this. These are cisternae. Then we have vesicles and vacuoles. Vesicles are small spherical structures and vacuoles are large spherical structures. The vesicles are found towards this face. Now these two faces, this is concave face and this is convex face. This convex face, this which is convex face, I'll not draw the arrow, I'll just write convex face of endoplasmic reticulum is also known as cis face, CIS, cis face. And this concave face, this is concave face of this is also known as trans face, TRANS, trans face. So we have two faces. One is concave face, which is also known as cis phase, and the convex phase and the concave phase, which is also known as trans phase. So we have two phases, convex phase and concave phase. Now, these vesicles, which are small, vesicles are these small spherical structures. These are vesicles, and these are vacuoles. Large spherical structures are vacuoles. So these are vacuoles, and these are vesicles. This is the difference between the two. Vesicles and vacuoles. The vesicles are found near the cis. I'm drawing few, and the vacuoles are found here near the trans phase, like this. The vacuoles are found near the trans phase, and the vesicles are found near the cis phase. This is the difference, and both are secretory in nature. Now, another thing, if this is a cell, this is plasma membrane, and this is nuclear membrane. This is nuclear membrane. This cis phase, this trans phase, which is concave phase, this is towards this side. That means it will be towards the plasma membrane side. If we make the Golgi complex like this, this trans phase is towards the plasma membrane, and this cis phase is towards the nuclear membrane. This is another point. Could you understand this point? The cis phase is towards the nuclear membrane, and the trans phase is towards the plasma membrane. Could you understand this point? Yes, is it clear, this point? The cis phase, okay, fine. So I'll write also here. Trans phase is towards plasma membrane, and cis phase is towards nuclear membrane. Are located near cis phase, whereas vacuoles are located near trans phase. This is another point that we are looking at. Okay, now another point is these cisternae. I'll mark here what are these. These are vesicles. These are vacuoles, and these are cisternae. This is their labeling: cisternae, vacuoles, and vesicles. And which side they are? This also I have told you. Now I want to tell you what is the number of cisternae in plant cells, animal cells, and in fungi. So the number of cisternae are maximum in plant cells. Cisternae, these tube-like structures, unbranched tube-like structures, cisternae. These cisternae are maximum in plant cell, maximum in number in plant cells. They are maximum in number in plant cells. That is, their number in plant cell varies from 10 to 12. That is 10 to 12. In animals, they are less than that. In animal cells, they are about say 5 to 8. And GC or GB is present. This is their function. This is their number. Okay. So that means when only one is present, we call it unicistered. And these are multicisternae, but more are in the plant cell as compared to the animal cells. Animal cells are few in number. So children, this is clear. Otherwise, if this is not clear, I can repeat for you, or I'll move to the next point, which are functions of Golgi complex. Yes. Is it clear? Anything in this which you could not understand? The board is blurry. Rohan, last point, repeat. Okay. Repeat last point. Okay. Is the board blurred for everybody? Is the board blurred for everybody, children? The other Ani is also saying yes. Then okay, I'll just check it. Sometimes this is because of the network issue. Now, is there some difference? Yes, beta. Is it better now? Yes, bache. Is there any difference now? It's the same, blurred, still blurred. Bache, is it still blurred? Okay, the picture, it's still blurred. Yes, ma'am. Siksha, you can read properly. Okay, it's blurred. What is it saying? It's blurred. It is saying poor connectivity. What is it saying? Just write again. Okay. So it can be because of the connectivity, beta. Okay, bandwidth is low. Fine. Just I'll stop it for a minute and then I'll start again, maybe because of that. Okay. Okay, children. But could you understand what I wrote on the board? Because I was speaking and writing. Could you understand what I wanted to teach you? Even if it is blurred, could you understand what I wanted to teach you, beta? Yes? Okay, you could understand, all of you. Everything is clear. Now is it okay? Understanding is clear, or beta, the written part is also clear now. I'll just bring it back to Yashika. Both are clear. Good, Yashika. Okay, now is there some difference, beta? Or still it's blurred? Still it's blurred. No, it's same. But it can be because of poor connectivity. Shall I join? Shall we join again? Shall we join again? So it will make some difference. I think we should join again. Fine? Okay, beta? Okay, I'll leave it and then I'll join again.